All this is Dr. Mobeen Sayed from drbeen.com. Welcome to one more show. So, as promised, today is the detail for the discussion about the Pfizer vaccine becoming a DNA or reverse transcribing into a DNA. I had done that discussion on this, I believe, Friday night. Today is the detail. After this lecture, I will connect or link that discussion to this video as well. So, with this, let's start. And also, let's do this thing that the questions about that study, I saw there were tons of questions. Let's use Rumble today for our chit chat and let's have the questions there. I may have some technical issues going to Rumble and trying to do the live for the first time. And I would communicate with you through the YouTube, but let's get through this one and then we will decide about that. So here is the study, intracellular reverse transcription of Pfizer-BionTech COVID-19 mRNA vaccine in vitro in human liver cell line. So quick look at the references. So this is drbeen.com. In the description of this video, there is a link that has amazing discount on drbeen.com. The price of one video is actually should be more than the price for 1,000 videos for which the link is there. So take advantage of that while that is there. This is drbeen.com. Here is the study. We'll go over this study today, but more in detail. Here are some food for thought studies. All of these links are in the description as well. We'll look into this near the end of the study when we have a critical part of the discussion, and that is, what does this DNA actually do? So time evolution, this is another study, this is another study, and more. So all of these studies, and this is the fluorescence technique, and I put that here as well. So let's start. So today's discussion is the detail, the process that they did, and the results that they saw. It's a deeper dive, it's a more scholarly discussion. So here is what they did. They took cells, human hepatoma cells, or human hepatoma 7 or HUH7 cells. They put those cells in 37 degrees centigrade, 5% carbon dioxide, 1% penicillin and streptomycin so that no other infection occurs and their samples are not tainted or destroyed. Then they have a DMEM medium. So of course the cells, if you culture them, you have to give them food. So this is that medium. Then what they did was that this one, imagine this one little, they call it pellet or a well, this little one plate. On that plate, there are 200,000 cells. So they would call it 200,000 cells per well. And then there were 24 wells. So this is the discussion here in this part. So cell culture, this is what I just presented. Now on to real-time RT-PCR. Then what they did was, once they had the cells cultured, they then took the vaccine, the Pfizer-BionTech, they diluted that vaccine with 0.9% NaCl. They brought it to the exact concentration that they wanted to test with, and they created 0.5 milligram per milliliter, 1.1 microgram, not milligram, 1 microgram per milliliter, 2 microgram per milliliter. And then they put those various concentrations on various wells. They incubated this whole thing, these cells, with or without vaccine. So on some cells, they had put the vaccine. Some cells did not have the vaccine. Those cells that had the vaccine, over there was 0.5 or 1 or 2. Then they kept those cells in the presence of the vaccine or absence of the vaccine as a control for comparison for 6 hours, 24 hours, and 48 hours. So what are they trying to figure out? What they were trying to figure out is that if they put this vaccine on the cells, what will happen to the vaccine? What will it do inside the cells? What will it do in 6 hours? What will it do in 24 hours? What will it do in 48 hours? And they also had a control where the vaccine was not in the cells. So they would say that if the cells are sitting by themselves without any vaccine, what is happening in them, so that they can compare the activities and understand what activities were because of vaccine, because of the concentration of the vaccine, and because of the time versus not vaccine. So that is this part. So if you see here, Huh7 cells were incubated with or without BioNTech vaccine. Then is a real-time RT-PCR. After this, when the cells sat with the vaccine, their question is in their head, will this vaccine enter the cells? If so, how efficiently? One. Second, once inside the cell, what will this vaccine do? Will it create DNA? If so, how much? How fast? So these are the kind of questions that they're trying to answer. So if you see here, the next step. So after 6 hours or 24 hours or 48 hours and various concentrations, they opened up the cells, right? And they extracted the material from the cell. They extracted RNA, they extracted DNA, and they extracted a very specific RNA for the cell, which is called line 1 RNA. And because today we are doing a discussion which is in detail, why were they interested in line 1? Line 1 is a special protein which is present usually in dividing cells. And remember, these are human hepatoma 7 cells, these are cancer cells, so they are by nature dividing. In us normal people who do not have cancers or in non-cancerous cells, our body is composed of two type of cells at the bigger level, division level. Those cells that are dividing, for example, cells in the bone marrow are dividing, immune system cells are being produced, blood cells are being produced, so these are dividing cells. Then cells that are dividing in the GIT, our GIT surfaces are always getting replenished. Cells of the skin, even the respiratory system cells, and so on. You can actually, sometimes when you see somebody going through chemo and you see the kind of issues that develop, they get immune suppression because the chemo would suppress dividing cells. So because immune cells are generated by division, that immune suppression occurs. They get GIT problems because GIT cells are replenished all the time. And when the chemo stops the GIT or reduces the division of the GIT cells, because chemo is trying to reduce the division of the cancer cells, it would just reduce division in all cells. So patient would develop GIT issues. Similarly, they would develop skin issues. That is why they lose hair, because the skin cells are not replenishing correctly as well. And similarly, they'll develop other issues. So same thing over here. These are hepatoma cells. These are dividing cells. Now, inside these cells, there is a special protein called line 1. Line 1 protein has a special role, and that is it helps with the reverse transcription. Reverse transcription means to take an RNA and convert that into a DNA. There are some viruses that actually have their own enzyme that can take their RNA and convert that into DNA. Here we are looking at a human protein that can take up possibly SARS-CoV-2's RNA or this vaccine's RNA. This paper is about the vaccine, not the SARS-CoV-2. So take up the vaccine's RNA and convert that into DNA. So the question is, will it do that? So to answer that question, researchers are first going to see, will the vaccine cause an increase in line 1? If it is going to cause an increase in line 1, that means there is a possibility that when there is more line 1, that might cause these DNA creation as well. So here, what they did was, they took those cells and they extracted the RNA. There were two types of RNA they were really interested in. One were the RNA of the vaccine itself. and the second one were the RNA for the line 1. So here you can stop and say, well, we are talking about DNAs. All of a sudden now we are talking about RNA for line 1. What is this mess? So it is actually very simple. To create the DNA, we need the line 1, for example. Line 1 is a protein. To generate line 1, we need the recipe of the line 1, which is the DNA gene. So let's say here this is a nucleus. Inside the nucleus, imagine that there is a gene for line 1. We need to express that gene. We need to create messenger RNA from that gene. Remember, it's a recipe. So when we take the recipe book, that is DNA's genes, and we make a copy of that recipe, that is RNA, then we bring that RNA outside of the nucleus, inside the cytoplasm, cytosol as well. We have ribosomes that will connect with this RNA and make the protein. So if we want to make line 1 protein, we have to first open up the genes for line 1. We have to then make a copy of the recipe in the form of messenger RNA for line 1. That messenger RNA should come out from nucleus into the cytoplasm. In the cytoplasm, it should connect with ribosome, and line 1 protein should be formed. Because they are trying to figure that all out, they are trying to see, is there increased level of line 1 protein or RNA first, not protein first, but RNA first, compared to those cells which do not have the vaccine on them. So that is what they did. They picked up those cells now, and they started looking for RNA for line 1, for vaccine itself, because the vaccine RNA is present too, and some other proteins as well, or other RNA as well, which are called house cleaning proteins. Their RNA as well, just to compare. So if all the cells activity is just increased, then the house cleaning activity would increase as well. But if the house cleaning activity is not increased, that means cells is functioning at normal levels, but line 1 is increased. That means the vaccine is kind of triggering the production of line 1. This is a very delicate and very important point. You would hear some smug researchers who would say, well, line 1 is produced all the time, and especially we're talking about hepatoma cells. Over here, the line 1 will be produced because they're always dividing. But if you look at the researchers were not that dumb. What they did was, they knew that line 1 would be greater. They added the vaccine, and then they saw, will line 1 increase from the baseline? So if there are some more hepatoma cells here, there is no vaccine on them, and they produce, I'm just going to make up a number, they produce 10 line 1. Then if you put vaccine on them, will they still produce 10 line 1 or 1,000 line 1? That's what researchers were trying to do. So they would pick up those RNAs, then they would amplify them. That is the PCR's function. It takes pieces of RNA or genetic material and just makes copies of it. So they amplified those RNAs. Then, once they had amplified the RNAs, now they wanted to understand, from these RNAs, will the actual protein will be formed as well? So if I go back here, if you would read this afterwards, so if you see here, next step, immunofluorescence, staining, and confocal imaging. So here now, they're saying, well, if the vaccine is there and is causing increase in line 1 expression, that makes more messenger RNA for line 1. Will that increased messenger RNA for line 1 now make increased line 1 protein? I'll keep repeating. DNA gene is expressed. It will make messenger RNA. Messenger RNA would come out in cytoplasm. It will go and make protein. So now they're trying to figure out, is there protein as well? So to figure that out, how we do it, I think in the COVID time, we all now know that if you want to figure out some protein, we can make antibodies against it. So they had some antibodies that were against the line 1 protein, and there is a special part in the line 1 protein called open reading frame 1. It actually has open reading frame 2 as well. But open reading frame 1 is interesting because open reading frame 1 protein goes back into the nucleus. And over there, it helps with reverse transposition and possibly reverse transcription. It can do that outside in the cytosol as well. So remember, please, cytosol is the cytoplasm outside of the nucleus, and then nucleus. So we got the protein, line 1. How much protein do we have? Do we have open reading frame? Is that separately present? Is that gone to nucleus? Is that present in the cytosol? They want to measure that all. So how do you measure those specific things? You make antibodies against them. Then you attach antibodies to these things. Then you do fluorescence imaging and you take pictures of the colors. And these antibodies can give off various colors when they become bound. And so those markers, the color, the signals can then be picked up. So what they did was they took those cells. Now they had 40,000 cells per well on one plate, 40,000 cells. These are those cells that they had vaccine on. Then they put those antibodies on them. Then they took pictures, fluorescent pictures. And that is why I had this little video in here. These are the kind of machines that do it. So these little slots that you see here, these are called the wells. Inside these wells, if you see here the very first diagram or picture, the wells are open. Then in the second one, they're closed, and then they're open again. So this all is actually done with these machines. So these cells, if you see carefully here or closely, I don't know if it is evident, on this slide here is this little blotch. That is actually cells. So the machine can bring the cells, put various reagents on it, antibodies on it, then it can do some culture for those things or keep them incubated for some time. Then you can wash them in this machine, and then you can start visualizing them. When you visualize them, they look something like this. So you stain them with various antibodies. So now if I go back here, so when I say that we take pictures, this is that kind of a machine that will take pictures. Good. Then what did the researchers do? They went to the next step and said, okay, now we've gotten the RNA, we will measure that. Now we've gotten the proteins data. Did we actually have the DNA as well? So this one here is an antibody against the DNA, and this is an host antibody. This antibody or this material can help identify pieces of DNA. So now they're looking at RNA, they're looking at proteins, they're looking at DNA. Then how do they measure? Do we have RNA? Where do we have RNA? And so on. So imagine this is a liver cell which has the vaccine in it. Then they have put antibodies on it to figure out line 1 proteins, open reading frame 1 protein, DNAs. What will happen is these things will glow because you have attached antibodies to them that are immunofluorescent or that can become shiny when they are bound. So now what they do is they take this whole cell's area and they measure the total fluorescence from it or total light coming from it. I'm using light is a very simplified way of saying it. They'll see the total light that comes from it. Then they'll see the total light that comes from the nucleus. And if you subtract the light from the nucleus from the total light, then you'll get the light from the cytoplasm. That is how they figure out how much of the material is out in the cytoplasm versus inside the nucleus. Does it make sense? And for those of us who are researchers or who are more interested in a little more deeper dive, they would know this, that staining intensity of the nucleus area, they measured that. And how did they measure it? You can say, how did you just know that this is a nuclear area in this cell? So what happened was this Hoest stain, if I'm pronouncing the name correctly, this stain and the boundaries of that stain identify the area of nucleus because this stain goes in the nucleus. And then the border of the fluorescence for line 1. So line 1 is present everywhere now. Let me repeat this. Line 1 is a protein that is produced. That protein, after production, some of it will be living in the cytoplasm. Some of it is going to go in the nucleus. We're not talking about the DNA, we're talking about the line 1 protein and open reading frame 1 specially to be translocated in the nucleus or present here. So if you see the boundary of the open reading frame 1 or line 1 protein, that boundary would cover the whole cell. And they can measure the intensity of the light within that boundary. That would give you the total cell's intensity. Then you look at the Hoest stain and look at that boundary, and that would give you nucleus. So then if nucleus is B and the whole cell is A, you subtract the intensity A minus B and you get cytoplasmic intensity. What is it that they did over here? What they did was they wanted to see how much of the line 1 open reading frame 1 stayed within the cytoplasm and how much of that went in the nucleus. We're not talking DNA, we're talking about line 1. So they measured this. Why did they measure it? Because it is interesting to see if the open reading frame 1 went into the nucleus, because if it goes in the nucleus, it can then help with the transposition, and if it is outside or inside, it can help with the reverse transcription as well. So they wanted to see if this activity is happening, and if that thing has gone in the nucleus, it would go and actually do some more actions as well. So then what did they do? Then they looked at this DNA purification. So now they know the RNA sizes or volumes, they know the line 1 proteins volumes. Now they want to see the result of all that, the DNA itself. And of course, there is the cell's own DNA, and then there is the DNA of the vaccine generated DNA. So, how do you separate them from each other? First of all, they open up these cells or they process these cells and they extract all the DNA out of them. Just like vacuum, you pick up the dust. So, how do they extract the DNA? There are DNA extraction kits. There are various molecules that can bind with DNA and kind of separate it out. That's one. Now, the problem is that if you extract the genetic material from a cell, there may be RNA mixed in there as well, because RNA is also genetic material. So, how do they remove RNA so that we are only dealing with DNA? What they do is they have enzymes called RNases. Whenever we use the word ases, that means something that would break down something. DNase mean breaks down DNA. RNase means break down RNA. Protease means breaks down protein. So here we have some RNases. Now they put those RNases on this material so that the RNA is removed. RNase's job is to pick up RNA and destroy it. So now the genetic material is removed from the cell. That includes any genetic material in the nucleus and any genetic material in the cytoplasm, because we don't know really the vaccine generated DNA. Where is it? Is it in the nucleus? Is it in the cytoplasm? So you just take all the DNA, wherever it is, and then you remove the RNA, and now you have at least the DNA. Now, from this whole huge DNA, which is our DNA plus vaccine DNA, how do they know where is the vaccine DNA? So for that, there is further working, and what they do is vaccine generated DNA on the paper should be 444 base pairs. Why? Because vaccine has a specific genetic material structure and length. From that, when the DNA is produced, we can actually see that it will be 444 base pairs. So what they do is they extract the DNA pieces that are 444 base pairs. Now you can say, fine, you have this long string of DNA, you divided it into 444 pieces, base pair pieces. How do you know that this is vaccine DNA or this is your other gene, some gene's DNA, part of it? So what they do then is that they use primers. Primers are like a template. Um, you know, like various mold. So if you have a mold that looks like my hand, it would only bind with something that would look like my hand as well, or that can fit my hand, correct? So that is called a primer. So they have primer for DNA sequence. That primer is then put on this segment that they extracted to make sure it is the vaccine DNA. So that is a verification process. But hold on. If you're a researcher, you would say, well, fine, you found some genetic material that is 444 base pairs, and that you're passing it on as a DNA. But really, that is the genetic material that is a copy of the vaccine's RNA. So maybe it is really just the vaccine's RNA, which you're passing along as DNA. So what they do is they take the vaccine's RNAs and separately amplify them, and then they try to see if it is the same thing as this DNA. So they have one more verification step. Then you could also say this, you can challenge them and say, well, the cell's own DNA may be doing this. This is not necessarily from vaccine. So remember, they have control cells. They have cells which do not have vaccine on them. So then they take the genetic material in them and see that do they have this material as well? If the control cells, the cells without the vaccine on them, also show this DNA, then the whole discussion is done. It's finished. DNA is everywhere. But here, they found that the DNA was specific in those cells that had vaccine on it, and they could verify that. So if you see here, the verification part, number one, they had 444 base pairs were cut out of DNA and was extracted using QI, a quick PCR purification kit. And then if you see here, look at this one, Sanger sequencing. So then they further amplified that DNA and verified it to be the DNA generated from the vaccine. And that was done by Sanger sequencing. So now they have everything. They have the pieces of DNA they're interested in. They have the protein that helps make them. They have the gene expression, and they have the genetic material. They have the vaccine. So what did they do? Now they start analyzing all of this data. They've done the experiment. Now they would analyze the data. So here is what they found. And now I'm going through their results. I'm just going to go one by one over these. First of all, they said, we found that the Pfizer-BionTech vaccine can easily enter liver cells. That lipid nanoparticle can enter the liver cells efficiently. Fine. I would suspect that to happen with any cells because that is what is the function of the lipid nanoparticle, to help cross cell membrane. So wherever you'll put it, it will go in those cells. So this is one that they proved. Then they said, number two, can we say that the human endogenous reverse transcriptor long interspersed nuclear element 1 or line 1 gene was expressed? Remember, we discussed it, that if the thinking is if you put the vaccine in a cell and it goes in the cell, it can trigger line 1 gene expression to make more line 1, which will make DNA. So they started looking in the nucleus and the RNA coming out of the nucleus for line 1. And they found that yes, RNA was found. That was for line 1, and it was found in greater quantities in the vaccinated cells. So if you go down here, this is what they're showing. This is control, this is vaccine. This is control, this is vaccine. This is control, this is vaccine. Or really control V1, V2, various doses of vaccine and so on. So now they've proven that fine, vaccine's presence triggers the gene expression of line 1. Okay, so now we know that too. Then it's a fine, now that line 1 gene expression has occurred, the RNA is coming out. Will that RNA actually become line 1 protein? So for that, they started this one, and they started measuring effect of BioNTech on human endogenous reverse transcriptase long interspersed nuclear element, and they measured that. See, they say next we studied the effect of line 1 protein level. This is actually very, very interesting diagram. I'm going to show it to you. So in this diagram, what they're doing is, let's look at the top line first. This very first box, the red color is the expression of line 1. And the first box where there is very little expression you're seeing, that is a control cell. Meaning this cell does not have vaccine on it. It is just liver hepatoma cell just sitting around eating food and having fun. And if you measure the line 1 in it, there is line 1, but not a lot. And this is where our smug researchers say, well, the line 1 will be produced in the liver cells. That is correct. In hepatoma liver cells, that is correct, because it is dividing. But what they would probably not tell you is that right here are the diagrams for when the vaccine is added, then the expression is increased. So really, the researchers are going to debate with each other to say, will the line 1 be produced in those cells of our human body which are not dividing? Muscle cells, for example, neurons. And you would find it in this article, the researchers say that there are stable cells. Stable cells are non-dividing cells of our body. There are stable cells like neurons that actually express line 1. That means if you put the vaccine on them, they would increase their expression. And then, of course, are very critical cells, immune system cells, GIT cells, skin cells. They also need to have line 1 because they're dividing. So we can't just say that, hey, in normal, healthy human being, there is no line 1, and the line 1 was only present in the cancer cells. So back here, this is the expression of line 1 in control. This is the expression of line 1 in the cytosol. Cytosol means outside the nucleus. At 0.5 microgram per milliliter concentration, this is 1 microgram, this is 2 microgram. If you see, there is not a lot of difference. All of these concentrations actually triggered the production of line 1 in the cytoplasm. Of course, from the gene, from the DNA, the nucleus, the gene expression of line 1 would occur, RNA would come out in the cytosol, specifically with the ribosomes and rough endoplasmic reticulum. That is where the protein will be formed. And so you can find that protein in the cytosol. Understood? Then the next question they had was, will the open reading frame go in the nucleus back, or will go back in the nucleus? So this lower set of pictures is actually nuclear image put on top of the cellular image, cytosol image. So if you see here, overlap of host, that is a nuclear image. And once again, here the very first one, some line 1 in the nucleus. Then at various doses, line 1 in the, or open reading frame 1 in the nucleus as well. This is beautiful diagram. And then they show the concentrations too. What did they prove here? In this step, they proved that the open reading frame 1 of the line 1, which is helping with the transposition and with the reverse transcription, is not only present in the cytosol, but is gone back into nucleus as well. This is a normal behavior. So, okay, we got that. Then what did they do? Then they said, okay, so we now know that the line 1 is there. And I want to clarify one thing in my diagrams. So here, this blue thing is the open reading frame. These red tails is the remaining line 1 protein. And they found that to be present in the nucleus and in the cytosol. The only clarity is they were measuring open reading frame. They were not measuring the whole line 1, although they have separately line 1 measurement, but these diagrams are for open reading frame 1. So now, was that the line 1 whole of it present in the cytosol, or just the open reading frame? They only talked about open reading frame. So if that is the case, this is why I put the question marks here. Then it would really be just the open reading frame. Does that matter? No, just for accuracy's sake, it is either line 1 complete or just the open reading frame 1, but still the function will still occur. Okay, now the next part, detection of the DNA. And detection of the DNA, what they did was, as I showed before, they had collected all the DNA from the cell. That means any DNA present in the cytoplasm and any DNA present in the nucleus, they collected that all. And then within that DNA, they looked for the vaccine generated DNA developed, and they found it. They found it to be present at 6 hours, at 12, 24 hours, 48 hours. They verified that by Sanger sequencing. Then they verified that by checking the RNA as well. So if you see here, they say we verified. Sanger sequencing confirmed that the DNA amplicons were identical to the BioNTech 162b2 sequence flanked by the primers. To ensure that the DNA amplicons were derived from DNA, but not RNA, meaning we were not taking RNA incorrectly as DNA. We also performed PCR on RNA purified from Huh7 cells treated with vaccine without RNase treatment, and no amplicons was detected in the RNA sample subject. So they said if we don't break down the RNA and just look at the same thing in that RNA, we didn't find this. So that means this was DNA. Now, the final part, which is interesting here, and I'm going to go to that food for thought here. Few things. One important thing that they left open is the following. They said, if I can just quickly search for you, integration. They left this question open, that was this piece of DNA integrated back in the genome, our genome. So they said, and this is actually a hot potato topic. This is a difficult topic to work with. And I would show you some studies where there are fights and debates that does it happen or not. And so I think these researchers were smart that they just kept themselves off of that part of the topic so that people can just look at what is here without challenging on a very different aspect. So here they're saying a recent study showed that endogenous line 1 mediates reverse transcription and integration of SARS-CoV-2 sequence in the genome of infected human cells. And I'll show you that study. Actually, let me show you that study right now. That is this one. Not this one, this one. Reverse transcribed SARS-CoV-2 RNA can integrate into the genome of cultured human cells and can be expressed in patients' derived tissue. This is Zhang et al. The date is May 2021. However, the whole discussion doesn't finish there because there is another article by, this is not a study, but this is an analysis. It's kind of a response. For example, sometimes I say, well, this researcher, what they're saying, but doctor, what they're saying is not really correct. So here, on 17 August, folks said, another set of Perry et al., they said, no evidence of SARS-CoV-2 reverse transcription and integration as the origin of chimeric transcripts in the patient tissue. So they're talking about the recent study. So if you see here, the recent paper by Zhang et al. Suggests a potential explanation of this phenomena by claiming that SARS-CoV-2 RNA can integrate into the genome of infected human cells. The authors also reanalyze RNA sequencing and then they continue. But what they're saying is there are reasons that their analysis was wrong. And because the analysis was wrong, of course, then the conclusion is wrong. And there is no evidence that this actually happens. I think because of this kind of a debate that is going on, these researchers deliberately just stayed out of this. And they said there are studies that show that there is integration. But for their own discussion, they say integrated. If I go to the next word here, at this stage, now this is authors, at this stage, we do not know if DNA reverse transcribed from the Pfizer-BionTech vaccine is integrated into the cell genome. So they stayed clear of it. They didn't take a side to say it is in there or not in there. Here is why. The work that they did, they measured the DNA, correct? So here, if I go back, here, they found the DNA from the cell. Now, if the DNA of the Pfizer-BionTech vaccine is in the cell, it could be in the cytosol, it could be in the nucleus, it could be in both places. We can say that it is not true that it is not in both places and somewhere else. That's not possible. Unless it is all contaminated and they were just faking it by adding the DNA segments by themselves. But I don't think that happened. So either the DNA is in here, in the cytoplasm, or in the nucleus, or in both places. They stayed out of it. They just left it and they said, we found DNA. Where is it? We are not telling, or we are not doing that part of the discussion or that part of the experiment. So they came back and they said, at this stage, we do not know if DNA reverse transcribed from BioNTech is integrated into the cell genome. Further studies are needed to demonstrate the effect of BioNTech vaccine on genomic integrity, including whole genome sequencing of cells exposed to BioNTech, as well as tissue from human subject who received the vaccination. So they cleared themselves and they said, we would like somebody else to do the study to say, is this genome present in the cells, DNA in the nucleus, number one, and number two, if present, is it acting like a gene to make spike proteins or other things that gene does? In case of Pfizer-BioNTech, that would be spike protein. Then they said the third thing, is it being expressed on the cell surfaces as well? They left that all out. They brought us all the way to saying found the DNA, and then they stopped. So food for thought, questions. Where is the DNA actually present? This is like Schrodinger's cat. Where is the DNA? We don't know. But we are sure it is in the cell. Is it integrated in the host genome? We also do not know. And the only way they kind of gave us an idea is to just show us other studies. So food for thought, is this DNA transcribing? So if I go back here once again to the food part. So it is interesting to read Zhang et al.'s hypothesis. In support of this hypothesis, we found the DNA copies of SARS-CoV-2 sequence can be integrated into the genome of infected human cells. Then they say this thing which I have made green. They said here, because we have detected only subgenomic sequences derived mainly from 3' end of the viral genome integrated into the DNA of the host cell, infectious virus cannot be produced on the integrated subgenomic SARS-CoV-2 sequence. What does this mean? What they're saying is the recipe page is in the DNA, is in the nucleus, is integrated, but it is a photocopy, it is reversed. So if I pick up some page and I bring that page in front of you, I could reverse it as well. So right now, let's say this page that you're seeing. If imagine you have a recipe book in which all these words are reversed, you may not be able to then work with that. So that's what they're saying. They're saying it is present in the genome, but from 3' end instead of 5' end. Because of that, it is useless piece of genetic material. It cannot be transcribed. It cannot be converted into RNA. That's what they are saying. So what are Zhang et al. Saying? They're saying we see the genome being integrated, but we don't think it can help make spike proteins. The response to them came from the folks here who said, well, we think you're just wrong in your analysis. There is no question of integration. Then I want us to look at some more studies. This is about retrovirus. In the retrovirus, it's a different virus, right? The process of reverse transcription generates in the cytoplasm. So here is an example of a virus that does make the DNA and makes it in the cytoplasm. A linear DNA duplex via an intricate series of steps. Good. Now I want you to see this one. This is the nuclear pore blockade reveals that HIV completes reverse transcription and uncoating in the nucleus. So this is another virus, different virus, not SARS-CoV-2. But this one makes its DNA inside the nucleus. So there are examples of DNA fragments being made in the cytoplasm or in the nucleus. Now, SARS-CoV-2 vaccine fragments are present, that is proven. Are they in the cytoplasm or the DNA nucleus? That is left out. I will actually give you one more example. The adenovirus-based vaccines for SARS-CoV-2. The adenovirus actually goes and binds to the nucleopore. So if I make nucleus here, and these are pores, holes in the nucleus, this is the cell. The adenovirus, we have done this discussion in the past for the vaccine discussion, that the adenovirus will be attached to this nucleopore, and it would inject the DNA in the nucleus. And the SARS-CoV-2 spike protein DNA. So adenovirus-based vaccines make DNA. That DNA actually will be formed in the nucleus. So it is actually possible to have a piece of DNA. There is a vaccine, Johnson & Johnson or Oxford AstraZeneca, they do it. Or even Russian Sputnik vaccine, they do it. They are adenovirus-based vaccine. They make DNA segments for SARS-CoV-2 inside the nucleus. Okay. So back here. Where is the DNA? Don't know. Is it integrated in the host genome? No, but I have now shared some studies with you. Is this DNA transcribing? Is it actually making spike proteins? We don't know. Interesting thing, if you look at the original study, they say the DNA that they found okay, so I am missing it, and when I start searching while you are waiting, I become confused and I become blind. But over here, they would have, if I search for the word flank here, Sanger sequencing confirmed that the DNA amplicons were identical to BioNTech BNT162B2 sequence flanked by the primers to ensure that the DNA, so it was a complete DNA, meaning one complete piece that can function. So they saw that back here. Here they're saying that it happens inside the nucleus. So it could So now the question is it transcribing? We don't know. When does it decay? When does it actually degrade and go away, or does it not go away? We don't know. Maybe it goes away. Maybe it gets broken down second day, third day, fourth day. Maybe it is not broken down. Don't know. At least, so I'm talking from this study. The reason I'm being so aligned to the study is that I tried to give my own opinion at one point as well. And somebody said, well, this is not what authors are saying. So I said, fine, I'll just go with the authors. So this is what they're saying. And they're not saying if the decay is happening or not. But for anyone who worries, look, Johnson and Johnson, AstraZeneca, Sputnik, they make DNA fragments, especially in the nucleus. We survive them too. What is their long-term effect? We don't know. But at least we are seeing people who get that. So then, and this is what the authors say as well. They say the long-term effects. So if I go back here, so here, this is actually very interesting for me because the authors brought us all the way to the DNA present, but then they gave just the hints there. For example, in the BionTech 162B2 toxicity report, no genotoxicity nor carcinogenicity studies have been provided. So if we don't suspect the DNA to go in the nucleus and get integrated or just be present there like a little poison, if we don't expect that, then why are we talking about genotoxicity or carcinogenicity study need? So of course there is a inclination there, there is a concern there, but they're not articulating it, and I can understand why. They just don't want to get in that debate. In my opinion, they might come back and they say, well, we just didn't have resources to do the remaining part of the experiment. If we had the money, we would do it. Maybe. So no genotoxicity nor carcinogenicity studies have been provided. Our study shows that BNT162B2 can be reverse transcribed to DNA in liver cells in line Huh7, and this may give rise to the concern if BioNTech 162B2 derived DNA may be integrated into the host genome and affect integrity of genomic DNA, which may potentially mediate genotoxic side effects. If they're not worried about this DNA present in the nucleus, then they would not talk about genotoxic side effect. You can only cause genotoxicity if you're next to the genes, and genes are in the nucleus. So if they were just totally sure that this is all in the cytoplasm and nothing else, they would not bring up these points. So they do have that in the back of their mind. They're just not clearly saying it. Okay, so back here. This is also true that this is human hepatoma cell line, and I think I've beaten this point to death, that line 1 and human hepatoma cell is not just by itself dismisses the whole research, because they're showing that human hepatoma cell in its without the vaccine and with the vaccine has a different level of expression of line 1. And then if I search here, it's a neuron here. Look, I now remember the study by heart. They're saying the cell proliferation is also active in several human tissues, such as the bone marrow or basal layers of epithelium, the surfaces, as well as during embryogenesis. And this is very important for them. And they're saying that the fetus is forming. All the cells are new, they're all being developed. What if it goes there for the fetus? We don't know. So they're giving examples of where line 1 is present, and is therefore necessary to examine the effect of BioNTech 162B2 on genomic integrity under such conditions. Why would they talk about genomic integrity if the DNA piece they're not worried about might interfere with the genes? Furthermore, effective retrotransposition. So now this retrotransposition is a different concept. Retrotransposition is called jumping genes. That's a different topic. If you would like it, please tell me and I'll discuss that separately. But effective retrotransposition of line 1 has also been reported in non-dividing and terminally differentiated cells. Terminally differentiated cells means those cells that have become stable will not divide any further. For example, neuron cells, muscle cells, such as human neurons. And they have connected two studies here that prove or show that line 1 is actually present in these stable cells too. So saying and trying to dismiss this study by saying this is only the cancer cells and that is why this whole idea is mute or moot, that's not really interesting. And the interesting thing, the word moot actually meant discussable. This is an idea that is discussable. And it over the time, whenever you say this is a moot point, people would mean it is a discussable point, and then slowly people started taking it to be not discussable. Okay, so back here. Hepatoma cells, dividing fast, gene expression. However, cells in bone marrow do that, GIT skin, line 1 is expressed in stable cells like neurons. This is the discussion. So now, I saw a lot of questions last time. There may be questions here as well. There are two ways to approach this. I have the Rumble, and so I also wanted to discuss this. I have no political leanings for medicine. I don't know Rumble is right wing, left wing. So I am just trying to find a platform where I can present myself. I'm not doing politics, I'm presenting concepts. So if Rumble is something that would allow me, regardless of what type it is, I would go and check that out. If Odyssey is the one, the only thing with Odyssey I found difficult was going live with Odyssey and then having interaction with you all becomes challenging. Now, will that happen with Rumble as well? I don't know. For example, if I go in here and if, for example, I do this, Kim F says Rumble allows free speech. So I can click that and show it. I don't know when I connect with Rumble, will I be able to do this? I cannot do it with Odyssey. So it takes away a bunch of experience and interaction, which is the point of a chit chat. So there are two ways today. One is I come back live on YouTube. We do the chit chat and answer the questions related to this study. Then we wait for Rumble to pick it up. Rumble picks it up. It is auto sync. And then I remove it from here. So there is no YouTube God coming in and being upset. The other one is I just tried the Rumble today. So I'm going to hang up. You give me the comment. How would you like it? And then we do it that way. So in the meantime, please like, subscribe, and share. And if you would like to support this work, quite a lot of hard work, then there is a link in the description for drbeen.com. There are about 1,000 videos there, my prime content. And the price, you will be blown away with the price. So I always say it that the price is a single video's price can be more than the price that you have it there. So if you would like to get that, take advantage of that plan. In addition to that, if you would like to support this work, there is a link for buying me a coffee or becoming a patron or using PayPal. All of those links are in the description. I'm going to hang up and wait for you to say, continue chit chat on the YouTube today or try Rumble. If we go Rumble, what I'll do is this, I'll go start Rumble, then put the link here on the community part in the YouTube. You can also go and search for Rumble, my name, and then we'll do the chit chat. So your turn to tell me what to do. Bye bye for now.